Okay, good morning boys once again, and we have been discussing, debating, trying to understand the important concepts of the chapter molecular basis of inheritance. So in that, yesterday and before that, we have been taking classes and discussing about various important concepts in the chapter. And basically we said what is genetic material and how to prove it. We discussed many experiments, right from Frederick Griffith's experiment, transforming principle. Then we went on to prove the chemical content of that based on McLeod-Becatti experiment. Then we went on with the Hershey and Chase experiment. Then yesterday we discussed about Michelson and Stahl experiment, wherein the breaking of DNA molecule, that is DNA replication, is semi-conservative by using isotopes. One strand we used nitrogen, normal nitrogen with atomic number 7 and atomic mass 14. Another one we used nitrogen with 7 and 15, one neutron we added, so it became heavy. So with that, when the DNA molecule has broken vertically, we could see the three types of DNAs being formed after centrifugation. One is the lighter one, having both the strands N14, N14. One, it is the hybrid, that is N14 and N15 mix, isn't it? And based on their heaviness, sedimentation was there. And we could observe the DNA having one strand with N15 was at the bottom of the centrifugation. Now, we said that the very breaking of DNA molecule leads to formation of RNA. So we gave a title for this: DNA replicating itself, forming RNA strand is transcription. Correct now? So transcription is the process of copying genetic information from one strand of DNA into RNA. Right? Look at that. This is the DNA molecule. And yesterday we said two strands, leading strand and lagging strand. And we said one will be continuous strand, another will be discontinuous strand. And the discontinuous strand is discontinuous because of formation of small pieces of DNA. Those pieces are discovered by a Japanese scientist named Wakajaki. So appropriately we call them Wakajaki fragments, right? And we also said that DNA to break and form RNA, which is called transcription, there must be certain substances to be present in the nucleoplasm or in the cytoplasm. One is the enzymes, various enzymes that are to be there. Isn't it? We discussed that. And also we discussed there must be present of RNA primer. With the presence of RNA primer, the DNA replication starts and synthesis of new DNA also starts. And then we started talking about the Wakajaki fragments joining together and the direction. Now let us see, already we have said adenine forms base pair with uracil. Why? Because it is no more DNA molecule, it is RNA molecule, isn't it? We said the difference between DNA and RNA, if you recollect from your memory, nitrogenous bases, we said purine pairs with pyrimidine. And we said AT, adenine, always pairs with thymine. But in case of RNA, there is no thymine. In place of thymine, there is another nitrogen base called uracil. So always Adenine will be pairing with uracil in case of RNA. And yesterday's class we said RNA is of 3 types in the RNA world. Okay, one is the messenger RNA. It will be just like a railway platform, I said, with bogies. Just imagine a goods train comes and parks itself in the railway platform. How it would look like? So basically there is a thread with nitrogenous bases. That thread is the railway platform and the nitrogen bases are goods compartments. That is how the messenger RNA will be. And why is it called messenger RNA? It gives messages. What is the message? The genetic information. And second one is the rRNA, ribosomal RNA. Okay, that means there is a RNA present in the ribosomes, and that ribosomal RNA is actually responsible for the protein synthesis. And then third one is tRNA, transfer RNA. Remember I said the clove shape, lavanga shape, tRNA, transfer RNA or transport RNA. This facilitates movement of materials. That's why the name transfer RNA. So mRNA, tRNA, rRNA. 3 types of RNAs will be there based on their function. Right. Now we go to the next slide. Here you can see there is a DNA dependent RNA polymerase. This is an enzyme. So it is basically DNA dependent, but what is it doing? It is synthesizing the RNA. Polymerase means many units of monomers. So here the monomer will be nucleotide. So many nucleotides join together and form a polymer, RNA polymer. So to form that RNA polymer, there must be an enzyme called RNA polymerase. And what does it do? It catalyzes polymerization. And there should be direction. What is the direction? 53, 5 prime to 3 prime direction. So same thing you can show in the diagram. The template strand, look at that. 5 prime to 3 prime. And there are 2. One is the template strand, as you are seeing. 3 prime to 5 prime acts as template strand. Yesterday's class I told you what is this 3 prime and 5 prime. Basically it is the direction, orientation. And this 3 and 5, the numerals indicate the carbon position, carbon attachment. Okay. So 3 prime to 5 prime is the direction of the template strand, and reverse. We said complementary. Remember I gave the example of Jaugal Srinath and Prasad. Not our Prasad, pace bowler Prasad. Isn't it? Why Sandeep is waiting? Okay, is it locked room? Right. Let me see. Yeah. Okay. Now anyone can join. So basically when we say it is complementary strands, one is directed in the form of 3 prime to 5 prime, another is directed in the form of 5 prime to 3 prime. We call this as complementary copies. So when we say complementary copies, I said both Jaugal Srinath and Venkatesh Prasad both are pace bowlers. But then the way they bowl, one bowls in swinger, another bowls out swinger. So we say complement each other in pace bowling. So the batsman gets confused. One over he has to adjust with Srinath for the in swingers. One next over he has to adjust with the Venkatesh Prasad with the out swingers. So always, the pace bowlers of any country will be picked up in this way, so that the batsman in confusion gets out, right? So here the DNA strands also are like that, complementary to each other in terms of orientation or direction. Now, next, both the strands are not copied during transcription. we know that only one will be copied. And this is so because the code for proteins is different in both strands. Now, what is code? We will talk in next few seconds. It is basically, you must understand here, the nitrogenous bases, and they will be in the form of triplet. 3 nitrogen bases together is code. That is the code language for protein synthesis. And this complicates the translation process. If 2 RNA molecules are produced simultaneously, this would be complementary, as I said, to one another or each other. It forms a double-stranded RNA and prevents translation. Now, as far as we are concerned, we will restrict ourselves to transcription now. Okay? And I said, this is what happens. Now look at that, A, T, G. What are all these? Yes, Sandeep? Nitrogenous bases. And 3 nitrogen bases will be moving ahead, will be forming a group. That's why we call this code as triplet code. Right? ATG is one triplet code. CAT triplet code. GCA triplet code, like that. And just now we said it is 3' to 5' in template strand. Look at that. It started off with 3', third carbon, ending with 5 carbon. And what about the other one? 5' to 3'. 3'. When transcription takes place, now which is having transcribed or transcripted? If you see the bottom one, TAC, in place of T, uracil is coming, see, in that pink line. I hope you are able to see. Yeah. Wherever T is there, you just observe. T is replaced with U, uracil. But direction is same orientation, 5' to 3'. And this forms what we call mRNA. Why are you calling mRNA, Kadkol? Why is it called? Whatever genetic information that is stored in the DNA is transcribed to RNA. That's why we are calling it as mRNA. Means what was in store up to now in DNA molecule has come to RNA molecule. Since the information is loaded in this, messages are stored in that, we call it as mRNA. I hope it's clear. Right? And you can see the same, the template strand and the coding strand. That's why we are using intentionally the coding strand word. Strand, the thread. Okay? Now, further, overall, how this transcription takes place. What are the requirements? Now, basically there should be a DNA molecule, okay, and then there should be a promoter region, okay. First, to start that replication, DNA replication, there must be a promoter region. Let me expand that. Yes, promoter region. Then there is a structural gene, right? And then after the process, as the DNA molecule starts breaking itself, at one point of time there has to be end, and that end point is called a terminator. So everything begins and at one point of time ends. All of us know that. So the beginning is the promoter region in that red box, and the closing is the terminator region. And of course, just now we said DNA molecule should have 2 strands, a template strand and a coding strand with the direction. Template strand 35, coding strand 53, 5' to 3'. Right? Coding strand will be coding strand will be always, yes, 53. This is one of the questions in your NEET exam, particularly, okay? And yesterday your brothers have written the exam. Did you ask them? How was the paper? Just go through that. If you can get the paper, just go through it. So basically it is the segment of DNA between the sites of initiation is promoter region, and terminator region of transcription. It consists of three regions, as we said. First one is the promoter region. So it is the binding site for RNA polymerase. What is RNA polymerase? Just now we discussed. It's the enzyme that helps in formation of RNA, and it is located towards 5' upstream. Look at that. Now you must focus on coding strand. The promoter region is with the 5'. And structural gene, the region between promoter and terminator. See, where transcription takes place. See the structural gene. Basically, what is a gene? If you ask me, all biology boys, we give the answer as portion of DNA. Portion of DNA is gene. And when Kadkol says portion of DNA, he knows that it is basically three molecules. One is the sugar molecule, another is the phosphate molecule, I said SP, superpentapolice, vertical line, and third one is nitrogen base. And if these three molecules are together in a particular place in the form of a helix, that portion can be referred as gene. That much you should understand at present. Okay? And as I said, after the process starts and the DNA starts forming RNA, there has to be a time where termination should take place. DNA should not form RNA further. So that place on the DNA where termination is taking place is referred to as terminator region. So terminator is the site where transcription stops, located towards 3' end, okay, downstream. Now look at that. We said upstream it is the 5', downstream it is the 3'. You can see the arrow mark before 3'. So that is the orientation, right? And now it is the segment of DNA between the sites of initiation and the termination of transcription. So three sites. Further we are showing you. Look at that. The promoter region, then there is structural gene, RNA coding sequence, then terminator, time comes where the termination should take place, and the promoter region is the site that initiates transcription, and the terminator region is the site that terminates the transcription. Okay, clear? So focus on that sketch. You can see upstream and downstream of gene. So this is how the DNA will be virtually breaking off. Now, as I said just now, what is a gene? Gene is the functional unit of inheritance. It is the DNA sequence coding for RNA, particularly which RNA? mRNA. And there is something called cistron. Cistron is segment of DNA coding for polypeptide. What is polypeptide? What is polypeptide chain? Any idea? Yes, chain of amino acids. Very good. Chain of amino acids is polypeptide chain. And this happens during protein synthesis. So it is the largest element of gene. Normally, again, neat exam question. Cistron. What is cistron? As a functional gene having polypeptidal chain. You can say that, right? So basically, there has to be a cell, a living cell, as I said yesterday. In the living cell, there should be nucleus, and inside the nucleus, chromosomes, and inside the chromosomes, DNA, and portion of DNA is gene. I hope you got the clarity now. Basically, there should be a living cell, alive, cell should be alive, and already I said a cell will be alive when cytoplasm is present. The difference between dead cell and live cell is presence and absence of cytoplasm. And once the cytoplasm is there, nucleus, one of the cell organelles will be there. Inside the nucleus, nucleoplasm. Inside the nucleoplasm, chromosomes. Inside the chromosomes, DNA. And the portion of DNA is gene. And basically, chemically, if you analyze, a gene is nothing but combination of three chemical molecules. Deoxyribose, sugar molecule, pentose, sugar, we said. Remember? Second one is phosphate group. And third one is nitrogenous bases. And in the nitrogen bases, you know, purines and pyrimidines. And there is a base pair rule. AT at adenine combines with thymine and vice versa. CG or GC, cytosine, combines with guanine. So that should make the composition of a gene. But for the formation of that, there has to be certain properties. What are those? So structural gene in a transcription unit is of two types. Monocistronic structural gene, that is split genes. It is seen in eukaryotes. Here the coding sequences. Now exons, there should be two types of genes, okay, exons and introns. Now here exon means the one that are outside. Expressed sequences are interrupted by introns. Introns are the intervening sequences. So exons appear in a processed mRNA, whereas introns don't appear in the processed mRNA. So in monocistronic structural gene, we said the structural gene will be there between promoter region and terminator region, isn't it? So that structural gene will be of two types. One is the monocistronic, as the name says, one cistron, monocistronic. And the second one is polycistronic. Poly means many, many cistronic. And you can see the monocistronic structure. Exon, intron, exon, intron, exon, intron. Right? This is how the monocistronic gene will be there. And polycistronic, it is seen in prokaryotes. Here there are no split genes. Look at that. Now there is no splitting of this. Genes are present in a row, continuous. Polycistronic structural gene. Gene 1, gene 2, gene 3, like that. Okay? That is the difference between monocistronic and polycistronic. And in bacteria, like prokaryotes, synthesis of all types of RNA are catalyzed by single RNA. In bacteria, how DNA forms RNA, it is basically catalysis. Okay? And how this catalysis will lead to formation, there are 3 steps in this. Look at that. Now we are talking of transcription in bacteria. Remember that. Transcription in bacteria. Initiation, first step. Elongation, the chain gets elongated, and finally termination, closing. So transcription, DNA to RNA process has 3 steps in bacteria: initiation, elongation, termination. Now look at that initiation. Yeah. There has to be 2 things compulsorily must be present. One is the RNA polymerase, that is the enzyme. Okay? First substance, chemical substance to be present for breaking the DNA molecule to form mRNA specifically, or RNA we can say. Okay? There should be a RNA polymerase. This is enzyme. And this enzyme will be having a factor called sigma factor. So sigma factor and RNA polymerase will ensure the process of DNA breaking, DNA replication. Which place? At what place? In the promoter region. Is it clear? So once these two factors are there, the DNA in bacteria starts splitting. Then as the RNA polymerase and the sigma factor moves, the chain gets elongated. Look at that. We said elongation of the chain. But look at that, sigma factor is a must. And the third one, as the sigma factor and the RNA polymerase enzyme starts moving, a place comes where there has to be closing, termination, we say. So that reason where DNA stops forming RNA, in specific mRNA, is called terminator region. I hope you are following. Once this termination takes place, from DNA, RNA is formed, RNA polymerase and the Rho factor comes out. See, for termination, you must note down, there is a factor called Rho factor. You write it like P. But sigma factor, you write it like, I should say, alpha. Okay. So two factors are compulsory, along with an enzyme called RNA polymerase. For initiation and elongation, it is the sigma factor. For the termination, it is the Rho factor. So that is how you can see three steps: initiation, elongation and termination. So you must practice these diagrams. Soon after my class, whenever you are free, practice this. Transcription in bacteria. Don't get confused. In the examination, two we will be talking. Transcription in prokaryotes, that is bacteria. Transcription in eukaryotes. Not what we talked just now is transcription in prokaryotes. Example, we have taken bacteria. And in this, since your first terminal exam will be of MCQ, 100% he will ask you this question to confuse you. What is that? Sigma factor. Name the factor that is required in the transcription process in the prokaryotes for initiation, and intentionally will give Rho factor. Where is the Rho factor? Where is the Rho factor? Termination. Right? You must differentiate this. For termination, it is Rho factor. For initiation and elongation, it is sigma factor. I hope you made it clear in your mind. So soon after my class, practice this. Right? Next, we continue the same process, initiation and elongation. And as we said, the role. Here, RNA polymerase enzyme binds at the promoter site of DNA. Binding should take place. And second, this causes local unwinding, unwinding of the DNA double helix. And then an initiation factor, just now we said sigma factor, present in RNA polymerase initiates the RNA synthesis. This is what we discussed up to now. Look at that. Unwinding of DNA molecule. Yeah. Yes, Vivek. Please speak. Yes, their presence is a must. Sigma factor helps in unwinding of the double helix model of DNA. Rho factor helps in closing down, termination. Otherwise, there is no stoppage of transcription. No, unless transcription gets stopped, next step has to be taken place. No, translation. What is ultimate aim of us? Protein synthesis. Be it bacteria, be it any organism, including us. Ultimately, the aim is to prepare, synthesize proteins. If only transcription takes place, then our aim is not reached or achieved. That is the reason there has to be a termination factor, and the termination factor is rho factor. Is it clear? Yes, Karkol? Yeah. Now the same thing is explaining again. Initiation, the promoter region, RNA polymerase enzyme attaches to it, releases the sigma factor. Sigma factor will open the double strand. As it runs through, you can see, as it runs through, the DNA molecule gets unwinded and helps in formation. You can see that yellow thread coming out. That yellow thread is nothing but RNA. So this is how DNA molecule forms RNA in prokaryotes, right? So RNA chain is synthesized in the 5' to 3'. In this process, activated ribonucleoside triphosphates. These are energy molecules, we said. ATP, adenosine triphosphate, GTP, guanine, uracil triphosphate, UTP, cytosine triphosphate, CTP, they are added. Why? These are energy giving molecules. And this is complementary to the base sequence. Right, this we know already in the DNA template. Look at that. These are added. Okay, and you can see this. Already we said the termination factor, rho factor, binds to RNA polymerase and ensures termination, isn't it? There has to be termination. Look at that. So once the termination takes place, then only RNA molecule is formed. Hope it's clear? Next we will go to next class. We will talk about the eukaryotes. Is it okay? There's some video. I will put this in the WhatsApp group. You can see it at home. Right? Take care. Bye.